Okay, so it is time to welcome the audience back for the Mary Cartwright Lecture 2025. And I'm delighted to have with us today Weini Huang, a reader in mathematical biology at Queen Mary University of London. So let me say a few words about Weini before passing the floor to her. So Weini has an interdisciplinary background and obtained her PhD in evolutionary theory in the Max Planck Institute of Evolutionary Biology in Germany in 2012. She then moved to the UK, to London, to work at the Bard Center Cancer Institute, initially as a postdoctoral researcher, and then became lecturer in mathematical biology in the School of Mathematical Sciences at Queen Mary in 2018. Since then, she has been focusing on modeling interactions in different biological systems, including cancer evolution. Weini and her team are interested in understanding how diversity and population patterns are formed and maintained in natural and human cell populations. The team uses theoretical approaches such as stochastic processes, differential equation, as well as computational approaches to model those systems, bringing a strong connection between their theoretical work and experimental and clinical observations. Weini collaborates with experimentalists, with experimental evolution groups and cancer biologists, clinicians globally. And the topics of her research go from evolution of trades-off and resistance to viral infection in microbial systems, drug resistance in cancer, cancer immune coevolution, dynamics of extra-chromosomal DNA, and mitochondrial DNA dynamics in healthy liver. Today, Weini will talk about her work mainly on the extra-chromosomal DNA and mutation accumulations in tumors and healthy tissues. So thank you very much again, Weini, for accepting our invite, and the floor is yours. Thank you.
Thank you. Let me just share my slides. I guess you can see it.
Yep, all good.
Yes.
Good. Cool. Thank you. Thank you, Sarah, and thank you, Kira, and everyone for. It's my honor to present in this lecture. And I noticed that there are not so many biological talks in the LMS yet. Some of them are maybe 10 years ago already. So I hope for the audience is like a fresh, how do we match between the field? So today I will talk about, so I work in different systems, but I focus on talking the cancer evolution today. So you might wondering, like, what is the link of cancer and evolution? I think that's the first thing when the clinicians, they hear about cancer evolution. What is that? So it's actually, Britain is a great place to talk about evolution because we all know about the, Darwin. So what Darwin proposed is that in our nature that we have variations. So what nature selection do is just to make those traits that are slightly better than they will be spread in the population. So, but in Darwin time, they don't know so much about the genomics. They know there is some genetic information, but they don't know how this can be spread among the different species. But with more and more information, maybe you see these posters about life of trees. So people to instruct about genomic information, fossil information, what is traits, as what Palami talked about, that can measure physically. And then what they can do is that they can tracing which species are linked to which species, and they draw a tree about life. And that's actually everything about how evolutions bring this diversity that's in the natural populations. But what is interesting is that evolution is not just static, so it's dynamic. So if you zoom in, so this is a picture of a wet lab. They work on E. coli, which is a bacterial species that they measure this species in a very controlled environment for many, many years. And then they can see that if you look at the bar here, so the bar tells the frequency of a certain mutation strains, certain bacterial strains, how do they change over the time. You can see in the beginning is gray, so everything is the same starting in the experiments. After many, many generations, you can see a high diverse. Sometimes you have mutations shows up, take over the population. Sometimes they went extinct when they are initially expand. And then you can see period with diversity. In this particular example, you can see in the end, these experiments come with that there's a mutate that happens. There's a big bang of multiple different type of strands happening in the system. So it's very dynamic in a way. So, but evolution, maybe in terms about nature species, we think about how things adapt to the environment. But evolution is not always about adaptation, especially if we think about disease in human perspective. So evolution will make changes in the system. For example, this is an example of measurement of a 59-year-old male. So we believe is one of the senior authors of this paper. So he donated his bone marrow, and in the bone marrow, they extract some cells and they measure how many mutations these bone marrow cells had compared with the original cell. And then you can see that there are in the order of thousands of mutation already happens in the normal cells of a healthy person. So most of these mutations, they don't do anything for our body, so they don't change any parts of the cell functions. But some of this mutation will do. So this is what leads to disease. So this is a picture here. Again, they're taken from, think kidney cancer. So the patient has metastasis. So you can see M2A, M2B here is the metastasis stage. So what they do is that they take different samples from the primary tumor in the kidney, also take some samples from the metastasis sites. So they generate a tree again, looks like a life of trees. Instead of each end of these leaves is a species, here is a tumor sample. You can see that this diversity between the tumor samples made the tumor different and made also a difference between the metastasis site, which is the M, compared with the primary tumor. What make it even maybe more like a little bit frustrating in terms about this evolutionary process is that quite often evolution is also the reason why the tumor can relapse and become resistant to treatment. Here is a picture that when I first come to London, one of my collaborators gave me this picture and said, we need to model this system. So this is an ovarian cancer. So the y-axis is a marker called CA125 to measure how many tumor cells in the blood. So when the tumor cells died, some of the dead cells come to the bloodstream, and the clinicians can use this to measure the tumor burden of the cells. And you can see in this particular case, the patient started treatment in the beginning at day 0, and it reacts very well to the treatment. So the tumor burden drops down. So then the tumor comes back again. So the clinicians use a different chemotherapy again. So it works again, but it comes back again. And then they have to change to the third line of chemotherapy. You can see with the process, the tumor slowly lose the reaction to the current treatment. So this chemotherapy doesn't work for the decrease in the tumor abundance anymore. So this means that behind just a measurement about how many number of cells you have, the tumor itself changed. So this change is coming from the evolutionary process. So you would maybe think about, okay, if you can measure it, so why do you need theoretical models? But in the reality is that quite often you can't measure it in clinical studies. So in cancer biology, people might do some experiments in the lab, which Paulami talked about cell lines, which is that originally there's a tumor cell from a patient. So they take the tumor cell, make them immortalized, so allow the cell to continue divides. So they grow in the petri dish, which is cell lines, or they can kind of make some environmental controls, allow the tumor cells become what we call organoids, which they mimic the organs of the patients or mimic the organs of animal models. But these are in the lab, so we call it in vitro. And also people might take some tumors cells and inject it in lab animals like mouse. They put normally in the bulb of the mouse and measure how the tumor changes. So these are all in the lab. You can measure some properties. You can take the samples over different times to measure whether a certain type of cell with a certain type of trait or genetic mutations, they grow faster or not. But in clinic studies, quite often, maybe you would have the patient have the tumor being detected and they have a chance to get surgery. But after surgery, it's very unlikely that you get another sample. So in the surgery, maybe you have multiple samples, like you have a spatial sample from the primary tumor or metastasis that I explained before. But in the relapse, quite often that is not possible to do a surgery anymore, so there's no samples going on. So the question comes that you have a system that in clinical studies, there's no possibility to take dynamic measurements, and you could only measure in the lab. But what happens is that the cells, they don't behave in the same way in the lab as in the body. So there's a gap in between to see how could have some tools maybe can apply both to the lab and to also to some measurement in clinic, reflecting the same thing, and let you gain confidence what mechanisms you observe in the lab is also maybe the mechanisms governing the cancer evolution in the patients. So what we do this, we call genetic footprint. So genetic footprint is genetic informations that you can reflect the dynamics that you can't measure directly, or it reflects some evolutionary history of how the tumor grows. So there are some properties that you could use as a genetic footprint. So the first properties is that it need to be inheritable. So you, for example, you have a cell division. So inside of this is my figure of chromosomes. So you get a mutation, which is the red dot here. When this cell divides, the mutation can be inherited by the two daughter cells, like how Palami explained about the fixation of the mutations. So this means inheritable. And the second property is that you need to be stochastically changes over time. So which means it does not just happen once, but it happens in a stochastic way in the whole process, which can be used as a measure of time. So these two things make genetic footprints that maybe can use to reflecting those properties that you can't measure over time. So there are different genetic footprints. So yesterday I invited someone come to Queen Mary Mass to talk about methylation, but that's not what I'm focused today. So today I'm just talking about the two genetic footprint that I'm working with. So the first one is called point mutations. So point mutation, as Palami explained, that you have the cell. In the cell, you have a nucleus. Inside the nucleus, you have chromosome. In the chromosome, you have DNA double strands. In each cell division, these DNA double strands need to be open and be copied. And if you think about the number, one human cell has 3 billion base pairs in this string need to be copied. And one cube centimeter tumor sample have 10 to the 9 cells. So there's a lot of DNA copy happening. So the error rates for even for our normal person, normal cell is 1 per cell division and goes to the order of 10 per cell division in the tumor cells. So this is point mutations. Another thing that I would talk about is the ecDNA. I think Palami mentioned to you. So ecDNA is that when you have this DNA replication, sometimes the DNA strand will break. So they drop out in the cell and they formula a circular structure. And this circular structure is because they don't have what Palami explained about centromeres. So they can't be equally partitioned to the daughter cells. So they are randomly partitioned. So the randomness that differs from the chromosome DNA is they make the diversity between cells evolve much faster. So for example, if you have a mutation happening, and then you need to slowly spread in the population. But with random partition, then you can have big difference between two daughter cells in a much short time scale compared with the chromosome DNA. So actually, if you think about the ecDNA, they not just happens in human, they happens everywhere in all the species. So here I just listed some examples, which in biology calls model organisms. So this is a model organism in yeast, and in telecrest is a model organism in plant, and fruit fly is a model organism in animal, and then even chicken, which is a like model organism for domestic animals, they all see ecDNA and they have a function. But of course, the one getting most attention is the human cancer, because there's a consequence when this genetic error happens. So this is a paper published in 2020, looking at the biggest database of different cancer samples and the cancer patients. So there are, I think actually back in the 60s, people already observed ecDNA in the microscope, but they didn't know what is the use of it, meaning of it. Now, with the new technology, especially with bioinformatics, they can distinguish what genetic signal is in the ecDNA, what is in the chromosome. So they could analyze this big database to see how relevant this ecDNA is for patients, for tumor patients. So the statistic is up to 33% all the samples across any tumor types carry ecDNA. So it's a high correlation about cancer and ecDNA, and up to 15% when they already see them in the first diagnosis. And if you look at here, the red colors are the percentage of samples carry ecDNA. You can see there are especially high in certain cancer types, like in glioblastoma is one type of brain cancer, in sarcoma, ovarian cancer, and also in neuroblastoma is a children cancer type. That's why if you later see what we compare with, most of the data is come from this glioblastoma with the brain tumor. And what interesting they found out is that this ecDNA, because they are a piece of DNA break down from the chromosome, so they can break down in different areas. But what they observe, those ecDNA quite often carries a specific genes that are highly associated with driven to the cancer itself. So which means that it could be a byproduct that because the disease already happening, or it could be a driving force. So this is one key question we first want to ask. Is ecDNA is the reason drive the disease, or is the byproduct of the disease? Is it association, or is the causation? So what makes people cares about ecDNA is that this is a survival curve over time. You can see the red line is referring to the patient has ecDNA. This line is the lowest, which means that if the patient has an ecDNA in the sample, the patient actually has lowest the survival rates over time. So I hope I explained to you that we have point mutation, we have ecDNA, then we need some statistical properties to measure in this kind of evolutionary process. So I have different stochastic statistical properties that I will talk about in this talk. First is called site frequency spectrum. So site frequency spectrum is that if you look at this simple figure here, so each one is a cell, and then you have the chromosome, and then you have the mutation marked by colors. In the cell division, you can stochastically get some mutations. And if you look at the end of the population, so you want to look at about some statistic about these mutations. The site frequency spectrum means the number of mutations reach a certain frequency. For example, if you see here, I have 3 cells survive in the end, so the gray one means the cell died. And you can see that if I count how many cells have this red mutation, it's 3 cells. So it's reached the frequency of 1 here. And if you want to see how many cells carry the yellow or the other mutation, is only 1 of the cells. So it has the frequency of 0.33. So 1 of the 3 cells carry it. So you can do statistics for the whole genome, because the whole genome has 3 million base pair. And we said every division introduce around 10 mutations. You can see there's a huge amount of mutations that you can draw such a site frequency spectrum to asking how many mutations reach a certain frequency. And this can be a statistical properties to reflecting some unmeasurable dynamic properties in the system. Another one is related, it's called the mutation burden distribution, which means instead of look how, for example, this red mutation, how many cells carry it, you can just look at each cell, count how many mutations are in this cell. In this example here, this particular cell has 3 mutations. So it's in line here. I have 1 cell carries 3 mutations. And then I have 2 cells carry 2 mutations. This is called mutation burden distribution, counting how many mutations in each cell. But this distribution requires more information, you can see, because this means that you need to know the information of every single cell. But with the site frequency spectrum, you just need to know the whole population. So you don't need to know which mutations are in which cell, you just need to count. And then another property that we use a lot is the ecDNA copy number distribution. So instead of looking at the chromosome here, I look at these circular structures and I count how many circular structures a cell has. So in this particular example here, I have these two cells has no ecDNA, so it's 0 copies, and have this one cell that has 2 copies. This is the copy number distribution. So this is the 3 properties that I will talk about. So the question is that, can we use genetic footprint that I explained to you to identify ongoing selection, which means some cells divide faster than the others, which is the aggressive a certain type of cells. And if you can identify them, then you can look at the mechanism why these cells are dividing faster to distinguish from random noise. And can we use this to infer key properties that we can't measure directly? So the answer is yes. So my former PhD student, Crystal, so this is talk about the point mutations. He's found out this statistical properties, not only that you can use to reflect in the dynamics, they're also related because they come from the same stochastic process. So he used a dynamic matrix to quantify the mutation accumulation in the same evolutionary process. If you look at it here, so yi here, so I means the like time, event time. So at a given time, how does the population look like? So each column means a mutation, each row means a cell. So if you have a 0, it means that the cell carries that mutation. The cell doesn't carry that mutation. If it's 1, means the cell carry that mutation. So you can see that if it's gray, it means that these 2 mutations happens in the same cell division. So for example, if you have this cell divided to 2, if this daughter cell get 2 mutations at the same time, there will be a 2 ones in the same gray area. So you can see that this matrix that can be summed in the different way. So you can sum them in the column, you can sum them also in the row. And that means the different statistic properties that I just mentioned before. If I look at each column, so which means that in the sum of each column means in this column, it means mutation 1 are carried by 3 cells. So if I look at the statistic of the sum of the columns, this gives me the side frequency spectrum that I introduced before. If I look at the sum of each row, which gives you the blue color, it tells how many ones you are in a cell, which means how many mutations that you are in a cell. So this column, blue column here, gives you the mutation burden distribution. And then the purple one tells how many divisions a cell already experienced, so which is division distribution. So we can see these properties, they are from the same matrix, so they should be able to equal to each other if you sum up through the blue properties, or you sum up from the orange one, which is the column. So what's interesting is that we can write down the recurrent relationships. How does this statistical properties change over time? So this is very similar as Palaimi introduced about fixation of the mutations. But now we are not just looking at one mutation. We're looking at a stochastic process that you have like continuous mutation come to the system, and then you want to look at the distribution of all the mutations. So in this way, Sj means that a given mutation that are carried by the number of mutations present in J cells. So, and I+1 means that you look at next time step. So this change of the properties obviously is related how the events in the last time step, which is I, and you can change from J-1 to J, or you can stay from J to J, depends on whether the cell is dividing or not dividing. So this equation is written in the assumption where we have this birth-death process, where you can written as a Markov chain. So beta is the birth and the delta is the death. Again, every mutation, you have this like fixation probability. You can arising from the first cell, you go to the second cell until you reach in the whole population with n cells. And then you have these 2 observing states, which is at 0 and n. If a mutation gets lost, it doesn't come back in the same position. If a mutation gets fixed, all the offsprings will have this mutation. So you have this 1 transition probably to the same states. So in the case when we call about pure burst process, which means that beta is equal to 1 and delta equal to 0, so which means the cell is just dividing, and we can write down the recurrent equation in this way here. And we can solve the system and get a solution while reflecting a certain power law shape. So the solution looks like this. So instead of using J here, I introduce F, which is frequency instead of abundance. So J is the number of cells carry a given mutation, and F is the frequency of this mutation in the population between 0 and 1. So if we use the frequency instead of the absolute value, we can see that in the assumption, if the beta is equal to 1, the delta equal to 0, so which means the population is keep dividing, then the site frequency spectrum follow a power law shape. So what is this relevant to? For example, if you think about healthy individual, if you look at young individuals like children, they are still building their stem cell population. So there's very, very few cell deaths happening in the population. So most of the cells just keep dividing. So in that case, you will kind of expecting that the mutations in those individuals should have a shape of F2 minus 2. So, but if you can also, this is the case if you think about tumor cells, because if you have tumor cells, they just divide very aggressively. So compared with the deaths, the birth is much higher. So you could more converge to think about with this shape of F2 to the minus 2. But this results is actually already is a long-term results from the field. So there are different people using different methods. Many of them use Fock-Planck equation to derive the same power law shape, and they apply to cancer types as well. So this particular paper here in 2016, they used the power law shape to fit the mutations across the same database that I mentioned before about all the cancer types. They found 30% of the cancers has the mutation fits a shape of F to the minus 2, which means that this 30% of tumor samples, they only have one event happens in the patient, arising the first tumor cells. Afterwards, everything is just noise. So it's random drift in the population. So this is very important. This actually changed the view of the clinicians about thinking about the cancer, because most clinicians will think about the cancer is a process, you become more and more aggressive. So after the first events, you have other events happening. So you always have a more aggressive cancer types arising in the patients, make the patients come to the end of the life. But with this simple model of fitting how the mutation should accumulate in the simplest case and compare with the data, they reveal 30% of patients, they don't have a second event to make the cancer more aggressive than the beginning. So the first arising cell already have all the properties about the cancer. And so what we can contribute further with Crystal's work and also with my colleague Dudley Stark. So he worked in combinatorics. So he helped us to find out solutions that when data is not equal to 0, so which means you don't only have cell birth, but you have also cell death. So you have birth-death process. And we could still find out the whole, approximate the distribution and look at different shapes of the distributions. And we could also look at other distributions. For example, we can look at the mutation burden distribution, which has these solutions here with the purple color here. And we can use this division distribution to get the mutation burden distribution, which has not been solved before in the field. So which means that, as I was explained, if you need the mutation burden distribution, which means you need to know the information of each single cells. And this technology was not there before 2010, for example. So it's become like the future of the field. People already start to know the information of the whole cancer, then they know there's a huge diversity among cancer cells. So the new technology is deriving to understanding just the information of the whole population to the information of single cells. So we allow these results allow us to have a theory for the new data coming out to say, okay, if you have the information of each single cells, how does this reflect into the dynamics of the system? So Crystal did the work, and so he has the exact solution if there's a pure birth case, which is the purple, which is the colored one, or he have like approximate solution if there's a birth-death process when the death is not equal to 0. So what we can do is that we use our solution to fit with some data in the field. So this is example of 8 samples. So they're all healthy person. So which means they have like birth, death rates almost equal to each other. And they would have counted the mutations in the, so they do the similar experiments. So they take some cells from the patient donors and then measure the mutations in these cells. So they can find out how many cells carry how many mutations, which is the mutation burden distribution. So we can use our analytical results, try to fit with the data to infer the parameter mu, which is the per division, how many mutations that you gain in a cell. So you can see that the fits, we're still working in a perfect fit. You can see the variance is slightly larger than our analytical results, but we are really happy about how this work can be combined together, because mutation rate in person is not a property that you can measure directly. So now we can use our distributions to quantify this in a systematic way. So what we can do further is that we can look at how unobserved changes in the population can reflect in these properties. So here again, I come back to the site frequency spectrum. So I was explaining to you that we have the f to the -2 is in a pure growth population. And there's another result come out is that I didn't talk about details, is that if you have a birth-death process, which means the birth rate is equal to death rate, this polar shape goes to f to the -1. So we could see that already from our theoretical work, there's a transit. If you let the population grow and then they converge to a constant population, you'll see this transit of this site frequency spectrum from f to the -2 to f to the -1. And that's exactly what people see actually in the data as well. So these dots here are healthy tissue from esophagus from 9 individuals. So they range from the age of 20s to 60s. So the shaded color is the young people, and then the darker color is the 60 plus the donors. You can see the mutations site frequency spectrum in the people is really shifting towards from f to the -2 to f to the -1. And what interesting is that we know that what we see is always a sampling result. So you have the population of a person, but what you measure is a few number of cells of the patient. So what we want to know is that how the sampling will change this distribution. So then the sampling will shift the distribution. So the blue line is the whole population, and the orange line is the sampled one. So which means this statistical property, the site frequency spectrum, will impact by sampling. So when we do inference back about these mutation rates or aggressiveness of the tumor divisions, we need to think about how does the sampling play a role there. But what interesting we found out is the mutation burden distribution, which is counting how many mutations in each single cells. They are not so much impacted by sampling. So again, so the sampled distribution is the orange one, and the full distribution with the whole population is the blue one with a single realization, or the gray one is the average. So you can see the two distributions, they overlap very well with each other. If you do inference, either use the information of the whole population, or you use a few sampled individuals, like 1%, then you can see the inferred parameter, they overlap very well with each other. So which means that if we think about the practice, you think about applications that we won't use our theoretical results to infer those mutation rates or selection fitness in patients and try to give a quantitative statement. We should think about which property we use and which properties will be more impacted by sampling. So talking about that, we also notice in the process that this process is not always a continuous process. So what we calculate is expected value, but what people have in patient is always a single trajectory. So it's one event. We found out there's a discontinuous in this single events compared with the expected value. So which means that when we look at some deviations from the patient, from the expected value, we need to also further ask us, is this really true that this process is not agree with average expectation, or it's because there are some there's some difference between single trajectories and average behavior. So yeah, talk about this, I would give the last moment to talk about the ecDNA copy number distribution. So EC copy number distribution, as what I mentioned, is that it's not have any controls in the centromere. So when the cell divides, that the copy numbers is random partitioned. So I play a small video here that you can see. This is a cell here. Every dot here is an ecDNA copy that you can see in the microscope. This is a cell during division. You can see in the beginning that you have one cell, now you have two cells. If you look at the dots between the two daughter cells, they are not equal from each other. So if you think about just a stochastic process about random partition, if you have two copies in the original mother cells, and these copies become duplicated, in cell division become 4 copies, these 4 copies need to go to one of the two daughter cells. So if this probability go to one of the daughter cells is 0.5, then you will have a single peak distribution. If this probability is not 0.5, you will have double peak distribution. And that's what they try to measure in these experiments here. So they take the cells, let them divide to 2, and they counted how many dots between the two daughter cells to see whether the distribution is single peak or not. And this is the results here. This is different cell lines from different derived from different cancer types. You can see all of these cell lines give you one single peak. So this just confirmed the assumption that this ecDNA are random partitioned between the daughter cells. This is our fundamental hypothesis that confirmed by the experiments. So what we did is that we construct a stochastic process to do either mathematical analysis or computer simulations to after different stochastic properties of the system. And we can compare with experimental data, either from the cell lines in the lab, or we can compare with the patient. So as what I explained before, what measures in the cell line does not necessarily behave the same way in the patient. So with our theoretical tools, we have one way that we use the same analysis, same inference for data from the cell lines from the patients to see whether they reflect into the same thing. So what we reflect here is what we want to know is whether the ecDNA is a driving force or not, which we call positive selection or neutral selection. So neutral selection is the gray one, positive selection is the purple one. So if the statistic shows neutral selection, then it should go to the gray one, otherwise shows to the purple one. So the conclusion is that we found all data in the patients refer to a positive selection. So the cells carry the ecDNA has to have a fitness advantage, otherwise they can't be in the system. So I try to use this simple demographic equations to explain you why it is the case. So this is the case about equation for the neutral case. So now we have NKT here, which means the number of cells carry K copies. So in the population that you have N0, N1, N2, so 0 means you don't carry any copies, and 1 means you carry 1 copies. So you can write down the demographic equations. So how do they change over time? So, and then the gray area come from the random partition, because if you have like I cells, copies in the mother cells, then you have 2 I copies duplicated, need to go to the daughter cells. So this follows just a binomial distribution. So we writing down the equation for the neutral case, we writing down the equation for the selection case, and then we could try to test whether our equation works. So we could decouple N with rho, which N is the population, total population size, and K is the population that carry K copies, rho is the frequency. So we want to see that if we want to know how the dynamics look like, we want to calculate the mean, the variance. So it's better to work on the frequency instead of the pure abundance. So this allows us to decouple the frequency with the abundance. And we can also do limited expansions, if we look at the large limits with the k is very large, so instead use a binomial distribution, we can use just a normal distribution where we assume that the k is large enough, so the distribution is really continuous in space. So by doing this, we could calculate the copy number distribution, not solve it completely, but we have approximations that has this interesting shape. So it has a exponential power law decay. And we can use this to compare with the data in the patient and the cell lines and infer the parameters. So when we did this, so we have B, so the first 8 panels here is different cell lines again in the lab. So they are again from different, derived from different cancer types. But below D and E actually is the data from patient. So D is the patient who have glioblastoma, is a brain tumor, and E is the patient has a neoplasma, is a children cancer. So you can see the dashed line is our best fitted distribution derived from our kind of approximation results, and the dotted results are the patient data. So we could infer the parameter selection from all this data. They all infer a parameter that is much higher than 1, which is the neutral case. So which means that to have a distribution like this, you have to have the cells carry ecDNA that are selected. So they have a higher fitness for the cells not carry ecDNA. So they are driving force of the tumor, not just a byproduct of the tumor. So in the last 2-3 minutes, I want to explain you. The story is more complicated because Sarah was asking, is all the cancer types the same? The question is not. If you see our equations before, we always have this S here, which Paulami also talked about, is selection coefficient. And you can see that in our equations, we always use just one number. And this number is not a function of the K, which means the copy number. So this means we have assumption here. We assume the fitness of these cells, ecDNA cells, is independent about how many copies they have. You hear from Paulami's talk, this can be very complicated. If you express more, you have more copies, maybe you have different behaviors. So this is just assumption that we make it simple for our calculations. But is this true? So this is the question we want to ask. So why it matters? So it matters for treatment, especially for relapse. So if you think about the people who have ecDNA, so that they get it detected, so you can see the cells have different copy numbers in the cells, and they have treatment, the treatment works. So they mainly, for example, this treatment, they mainly work in killing these cells have high ecDNA copies. So they will change the distribution in the population to a different distribution after treatment. The question is that when the patient comes back with relapse, is the same treatment will work again for the patient or not? Obviously, because the treatment is started on the ecDNA, so they rely on the distribution of the ecDNA copy numbers in the population. So you want to know in this short time before treatment and the relapse, whether the population will recover to a similar distribution like before, or it will not. So if it will cover to a high copy number distribution again, then maybe the treatment will work again. But if not, if you only have this very small copy number cells, maybe the treatment will not work again. So this obviously how fast this dynamics happens matters a lot with whether the fitness function is copy number dependent or not. So to do this, that another PhD student from you, she did some simulations. So she has different fitness function for the copy number. So the orange one is the copy number independent, which assume S is just a number by itself. So the blue one is copy number dependent, which means S is a function of K. Or you have the neutral one that the S is just 1. So ecDNA or non-ecDNA doesn't matter for the cells. So you can see easily the average behavior or the single trajectory. If you have ecDNA copy number dependent, the mean copy number and increase very fast in short time. So when we talk with this, we thought about there's a simple experiments people can just do. So you can have a population, you let them grow, you take individual cells because you random sample them, so they might carry different copy numbers in these individual cells. You let them grow in a certain time, and then you measure the number of the cells in each curve to see what is the population size. If the growth is not related to the copy number, then no matter where you start, you should end up in a similar size in the end. If the growth of the cell depends a lot on where the copy number is, then you have a distribution of this clone size again. So when we think about this, that we can do this in experiments, one of our collaborators said we can do this in our lab. So then did exactly the same experiments in the lab. So do they take cells and then they have different shapes? But because they are both clinicians work in the hospital, so they also care about how treatment goes. So instead only put a function about copy number fitness in the growth, we also need to think about how death works. So we put another function assuming that either death of the cell and the treatment will relate to the copy number or not. So this is our simulation results. So this again in the left side, if it's the copy number is if the fitness is copy number independent, you should see a shift of the distribution of the final population size from relate to the copy number. But if it's copy number independent, which is the green color here, then you can see even you include stochasticity, the distribution is more or less in the same shape and correspond to the same X values. So this is exactly what they see in the lab. So again here, these results here in the right side is what they measure in the lab. You can see it agrees with the copy number dependent fitness very well. So this confirms that the dynamics of these ecDNA cells is very complicated, that if you want to apply these results in the clinical treatment, we need to think about a more complicated version just instead of constant population fitness. We need to think about also copy number dependent fitness. So come to here, I come to the end of my talk. So I should first thank my collaborators. In the right-hand side are all the theoreticians. In the left-hand side is all the theoreticians. In the right-hand side is all the clinicians that we work with. And thanks you for your listening, and thanks the group.
Thank you very much, Weini, for such a fascinating lecture and for working on such an important topics. I open the floor for questions. As Kiran said, you can either use the chat or raise your hand and unmute to ask a question. Let me check. I don't see further questions. So this brings us to the close of the proceedings. So let me thank again Weini for her lecture.